My name is Vraj Patel, and the lab I had was bacterial transformation. So the purpose of this lab is to transform the E. coli bacteria when it's exposed to the plasmid DNA, and the plasmid DNA contains the P green gene and the ampicillin resistant gene. What you need for this lab is a micropipette, inoculating loop, calcium chloride, glass beads, 4 petri dishes, and 4 test tubes. And you also need E. coli, plasmids, P green, agar, lurea broth, an incubator, ampicillin, hot water, ice, and tape and a Sharpie so you can label the petri dishes. So the first thing you need to do is mark a sterile tube plus plasmid, which indicates that it has the plasmid added to it, and another minus plasmid. It has no plasmid. So then you would add 250 microliters of ice cold calcium chloride to each tube using a sterile pipette. Then you would place both tubes in ice. Right, you need to use a sterile inoculating loop to transfer E. coli to the plus plasmid tube and make sure you don't get any agar. And once you get the E. coli on the inoculating loop, you put it into the tube and spin it. And then after that, immediately suspend the cells by repeatedly micropipetting the cells by going like that over and over in the tube. And then you would put the plus plasmid tube in the ice, and then you would get the minus plasmid tube and do the same thing again, and then put that in the ice. After that, you would use the inoculating loop again to add 1 loopful of plasmid to the plus plasmid tube. And you would again put the plasmid on the inoculating loop and put it in the tube and spin the loop. Then you would return the plus plasmid tube back into the ice, and you would incubate both tubes for 15 minutes in the ice. Label the petri dishes plus plasmid, minus plasmid, plus plasmid, lurea broth, and ampicillin, minus plasmid, lurea broth, and ampicillin. The minus plasmid is the control, the plus plasmid, lurea broth, and ampicillin is the experimental plate, and the minus plasmid, broth, and ampicillin is the negative control. After 15 minutes of incubation, you would heat shock the cells. So basically, you would remove the tubes from the ice and immediately put them in 42 degrees Celsius water for 90 seconds. And then you would return the tubes back into the ice for 1 minute. Once you did that, you have to add 250 microliters of broth to each tube using a sterile pipette. And then after you do that, gently tap the tubes. Place them on a rack for 5 to 10 minutes for recovery. You would use a sterile transfer pipette to add 10 microliters of cells from the minus plasmid tube to each appropriate plate. Then you would clamshell the lid and pour 4 to 6 beads onto each plate. Use a back and forth motion to spread the
You're at 6 minutes.
I gotta cut a lot of this out though.
Yeah, so you're about 3. And you're good.
Okay.
You should be just about 5 minutes.
Okay. Once you have the glass beads in the petri dishes, use a back and forth motion and shake the petri dish. And when you do that, let the plates rest for several minutes to allow the cell suspensions to become absorbed into the agar. And then after that, you would hold each plate vertically and clamshell the beads out. Add 100 microliters of cell suspension from the plus plasmid tube to each appropriate plate, and you would add glass beads into those and shake it as well. Then after that, you would stack all the plates, wrap them with tape, and put them upside down into an incubator or at room temperature. If you put them in an incubator, you need to keep them in there for 24 to 36 hours at 37 degrees Celsius. Or at room temperature, keep them there for 48 to 72 hours. Some source of error could have been that the bacteria could have been contaminated when the petri dishes were opened. The heat shock could have also been done wrong, or an improper amount of broth, calcium chloride, or cell suspension could have been used. The incubation could have been set to the wrong temperature, which would have also altered the results. Once you've done the experiment, get a UV light and look at the dishes under the light, and you would see that the plate with the plasmids and ampicillin would have some colonies, and the plate with no plasmid and ampicillin would have no bacteria growth, and the plate with no plasmid and just broth would have a lawn, and the plate with plasmid and just broth would also have a lawn. The materials needed for this project is a micropipette, inoculating loop. God dang it! Look, at no, move, show the people this. Show. You can't see me.
Alright, Vraj. Ready? 3, 2,